Hello, everyone. I'm Norman Baylor. I will be the moderator for this next session. In this session, we'll discuss the state of play of vaccines. We'll cover from a regulatory perspective, gaps, safety, efficacy, and a global perspective on the situation with H5 and pandemic influenza vaccines. We have two panelists who are investigators, two panelists who are regulatory authorities, are from regulatory authorities, and one panelist will give a global perspective from CEPI. So we'll start with Arnold Monto from the University of Michigan. Arnold, I pass it on to you.
Thank you. I'm going to give an overview of the situation. Can you see my slide?
Not yet.
Oh, dear. Okay, well, let me get back into previous mode for screen sharing, which I am trying to do as we speak. Okay, let's try this again. Okay, now do you see it?
Yet.
Okay, technology. I'm going to try to give an high-level overview of the situation as I see it, and take as background some of the issues that occurred the last time we had a flu pandemic in 2009 with an H1N1 virus. So this is looking at the phases in which we will be, in order to make the decisions about how to get vaccines developed and out. We're currently in what I would call a pre-pandemic phase. The virus is infecting animals with rare transmission to humans, no evidence of human-to-human spread. And here the goal is a limited one, to modify disease in infected humans by treatment, by prevention, and to prevent adaptation, which is the main reason that we're concerned right now about the current situation. And speed of production and availability, large-scale availability of vaccine is not critical. And if you have to inoculate more than once, an initial inoculation and a booster, that's not a particular problem. And that's where we are, as I see it right now, where we have in the stockpile vaccines that were developed basically in the 2005 era that you've heard about. And we can produce and continue to produce vaccines using this kind of formula. When you're in a pandemic, the situation is different. Human-to-human spread is occurring, and a lot, of course, depends on how widespread it is and how severe it is. And large quantities are needed as soon as possible of vaccine. Multiple inoculations are maybe necessary, but are undesirable. We can think back to the COVID situation, where some countries actually separated out the initial COVID inoculation to get some level of efficacy in a larger number of people. So low levels of efficacy may be acceptable on the population level because they'll help to produce herd immunity. So that is a different situation, and we'll hear about regulatory issues that apply to that situation. The stockpile of vaccines, as I said before, will be useful only for pre-pandemic and early pandemic use. They're in relatively limited supply compared to the population that we will want to inoculate. And that's looking exclusively at the US and not even paying a whole lot of concern, at least initially, to the rest of the world. The caveat to this is the vaccines in the stockpile may not be close enough to the circulating strain to give good immunity. They should be used nonetheless, because of the point I made before, that lower efficacy may be useful in developing herd immunity in the population. Now, what can be done to increase supplies quickly? And I'm going to just point out some lessons from 2009, because regulation as well as technology will make a difference in terms of leading the manufacturers, who are a key element in all this. In vaccine development, what to look at, how to increase production of things that are already in the pipeline, which is where we hope to be. And in the US, this was viewed as a strange change. 2009, going from the old H1N1 to the pandemic H1N1. And human testing still was required, because this was a different virus, not resembling one that was previously seen. The question was how much human testing was necessary, and in fact, there was a push to get the vaccine out. Many felt that it could wait for the winter season, because that's when flu typically occurs. And many of us pointed out that in a pandemic, you're going to have flu occurring at other times, not waiting for the winter season. And therefore, we were lucky, because the regular vaccine at the regular dose did work. And we had vaccine available in the early autumn of 2009, virus being identified in the early spring. In Europe, they had mock-up dossiers. They were already, from a regulatory standpoint, with dossiers for licensure. All they had to do was put in the details in terms of the virus. But this resulted in something that turned out to be quite unnecessary, using the same kind of approach that had been developed for H5, 2 inoculations, adjuvanted vaccines. And therefore, as we go forward, yes, mock-up dossiers are useful, but they need to be much more flexible to reflect what is being what is in circulation. So that's my view of the background, and I'll turn this back over to Norm for the next speaker.
Okay, Norm, ready?
Yes, thank you, Arnold. The next speaker will be John Treanor from BARDA.
All right, let's see if I can do this. Okay, so how's that looking, Norm?
It's great. Can you put it in presentation mode?
Okay, so this is always a thing, because it is in presentation mode, but I have to switch. There, how's that look?
There you go.
Okay, good. So this is really what Arnold already covered. The only thing I wanted to point out, in case people did not know that, is that there are currently 3 approved H5 vaccines in the US. The Sanofi vaccine, which was 2 doses with 90 micrograms for each dose, is approved unadjuvanted in 18 to 64-year-old. The CSL Seqirus cell culture grown adjuvanted with MF59, approved at 7.5 micrograms, 2 doses in individuals 6 months and older, and GSK 2 doses at 3.75 micrograms, adjuvanted with ASO3 in 6 months and older. There are active studies ongoing to use these approaches for the most recent H5 viruses and submit dossiers for those, as well as a study to try and see whether a Sanofi vaccine with adjuvant could be used at lower doses, and that work is ongoing. Next slide. Oh, what am I doing? I do the slides. Sorry. I'm not used to this. All right. So I was asked to talk about things to try and improve the response. And as Arnold pointed out, speed is very important. And that's probably the crux of most development activities for vaccines right now, is how can we make them faster, get them out faster? And when you think about this, it's useful to think about it from the point of view of 3 different buckets. There's the pre-pandemic bucket, the beginning pandemic bucket, and the pandemic phase bucket. So in the pre-pandemic, if you had a vaccine that could protect against all strains of all subtypes of influenza A, that could be part of the seasonal strategy, that you have a vaccine that is broadly protective against all influenza A viruses, then you would already be prepared for a pandemic because the population would be getting these vaccines that were broadly protective. Or if you had a vaccine that was specific for, let's say, H5, but which was designed to be able to provide broad protection against all H5 variants without a need to try and create something that was an antigenic match to an emerging pandemic, this would be something that could be administered at the very beginning of a pandemic without the need to generate a new matched vaccine. And then you have the pandemic phase where you accept that the idea will be you want to create a vaccine that matches the pandemic and try and get that out as quickly as possible. And when you think about all this, I think it's important to understand that vaccine design is really only just part of this whole response. There's all the other stuff that goes along with it: production, fill finish, distribution, administration, sustainability, all impact whether this is a successful response to controlling a pandemic. And along those lines, trying to look at ways to make these different types of vaccines, there are many, many investigational approaches that are currently being done all over the world. And this is just a partial list of some of the things that people have been working on. The one that's attracted the most attention recently, of course, are messenger RNA or self-amplifying messenger RNA vaccines, similar to what was used for COVID, because of the much faster ability to make these vaccines and get them out the door, because they're released and potency assays and all this kind of stuff is faster, and you can make them faster than you can the traditional vaccine. So an attractive approach to responding to a pandemic. Other ideas to try and make vaccines that target common epitopes on the HA, the neuraminidase, or the M2 that could potentially provide broad cross protection. Strategies to try and make H5 vaccines which would provide broad protection by being located in the center of the antigenic map. Ideas of stimulating other modes of immunity, like cellular immunity using peptides or other approaches, novel adjuvants, new platforms, new types of nanoparticles, thoughts about using live attenuated influenza virus vaccines, of which there are several in development that might be able to stimulate mucosal immunity that could be more effective at preventing transmission. And the idea that possibly there would be ways, for example, by using self-amplifying messenger RNA, to have vaccines would be effective with only a single dose, which again would improve that response ability. And then there's a lot of ideas about using alternative methods of administration with these novel vaccines, including dermal patches, giving vaccine by aerosol or nasal spray, oral tablets, or other forms of solutions that would allow mucosal immunity. And then many of these new vaccines are designed to induce novel immune mechanisms that are in addition to or different from what we typically are trying to induce with inactivated flu vaccines. So what are the obstacles, of which there are many? And this is just a partial list of things that people are actively looking at. The pathway to licensure, I know Dr. Kaslow is going to be talking about this. The data, being clear what's required, what is a strain change, how much different does something have to be to require a strain change, what are the most rapid pathways towards authorization of novel approaches during a public health emergency, something that looks very promising but has not been demonstrated to be effective for seasonal flu. Is there a pathway for using something like that during a PHE? And, general basic research to try and have a clearer understanding of immune mechanisms of protection, things other than antibody. How do we understand their ability to really provide protection? And can we develop more robust correlates of protection or correlates of risk? Right now, from licensing point of view, that's really focused on antibody that neutralizes the virus, or specifically hemagglutination inhibiting antibody, but there is an interest in whether or not it would be possible to develop other correlates of protection that might be useful in vaccine development, making progress in getting better assessments of mucosal immunity. This has always been one of the most frustrating things, particularly in the development of live vaccines, that it's been so difficult to establish a correlate of protection for something like flu mist, because assessments of mucosal immunity, which is probably what the mechanism of the vaccine is, are so difficult. Just how to collect the samples correctly, what are the best assays, trying to make that development. And then the other issue that I think people have brought up is this interesting idea about how does imprinting and priming affect the response to subsequent infections or vaccines. Pointed out that there were differences between people born before and after 1968 in their apparent susceptibility to H7 or H5 infection. And this may have something to do with the effect of your first exposure to influenza on imprinting your immune system to affect the way you respond to subsequent exposures. Developing improved models of influenza, and possibly the development of the controlled human infection model. And as people already discussed, enhanced surveillance at that animal-human interface, especially trying to understand what are the best predictors of risk that could be used to make a decision about whether vaccination should begin. We right now know that the virus is there. What's the signal that would tell us, yes, we need to start vaccinating people? And really nailing that down is another thing I think is an important part of the knowledge gaps that we have right now. So with that, stop and hand it back to you, Norm.
Thank you, John. So we've heard two discussions and two presentations about sort of giving us a backdrop to what we need to move forward in the sense of the vaccines themselves. And the next question would be, where are we as far as the regulators? We really can't move forward with any vaccine until they're evaluated by the national regulatory authorities. And so our next speaker is David Kaslow from the FDA, and he's going to give us a little backdrop on where the FDA is and the requirements.
Thank you, Norman. Can you see my slides?
Yes, just in presentation mode, we're ready to go.
Okay, great. So thank you, Norman. As agreed, I'll start with a very brief.
Can you put that in presentation mode? And
let me try again. How's that?
There you go. Thank you.
All right. Thanks, Norman. So as agreed, I'm going to start with a very brief reprise of the pandemic influenza strain change proposal that we reviewed at the October 10th VRBPAC meeting. And what's shown on this slide is the strain change process described by Weir and Gruber in 2016. And the concept was that building off of a U.S. licensed seasonal influenza vaccine for which there was demonstrated clinical efficacy, a manufacturer of a U.S. licensed seasonal influenza vaccine could license a subtype-specific prototype pandemic influenza vaccine, such as H5N1, based on clinical safety and immunogenicity, with the effectiveness inferred from the efficacy of the seasonal vaccine. Implicit in this model was that as the prototype pandemic vaccines were updated and additional safety and immunogenicity accrued with those updated prototype vaccines, a manufacturer's strain change supplement would suffice if and when a pandemic occurred. Because the review and approval of periodic updates to those prototype pandemic vaccines did not occur, and to take advantage of the interpandemic period to accrue additional safety and immunogenicity evidence with updated prototype vaccines, our thinking has now evolved to consider a model where accrual of that additional safety and immunogenicity evidence with the updated prototype vaccines is made much more explicit, as depicted in this red interpandemic period box. This proposed process has at least two advantages. First, it provides a larger evidence base for relying on manufacturing strain change supplements during the urgent response to a pandemic. And second, with ongoing interpandemic updates to the prototype vaccine, coupled with better and better tools to forecast effective pandemic vaccine composition, we may save critical pandemic response time by having the updated vaccines we need without waiting for a strain change. So with that model in mind, and in particular the leftmost part that applies to new vaccines, from a regulatory perspective, what are the scientific gaps? And to start to answer that question, let's take some scientific gaps off the table. As noted in our current guidance on the clinical data needed to support the licensure of influenza vaccines, our long-standing guidance for industry on the content and format of chemistry, manufacturing, and control information and establishment description information for a vaccine or related products makes, I think, quite clear what the CMC expectations are. While there may be some CMC technical gaps to be addressed, typically by the time a vaccine candidate has made it to clinical studies, the CMC scientific gaps have largely been filled. Maybe two caveats there, one of which is really a truism, and that is product potency assays are almost always a CMC Achilles heel. How much of a scientific gap exists in potency assays may largely depend on the manufacturing technology and may be highly product specific. A related caveat is if and how implementation of the nascent Platform Technology Designation Program, which was recently established by Section 506K of the Federal Food, Drug, and Cosmetic Act, might facilitate some streamlining for manufacturing technology that meet that program's eligibility criteria, which includes prior approved products manufactured with that platform technology. So with that said, what we're really left considering are what are the non-clinical and clinical scientific gaps? And in the interest of time, I'm going to focus on what I think is the most challenging of the two, which is the clinical data needs. Again, it might be good to start by stating that the safety data needs are well spelled out in several guidance documents. So other than the scientific gaps and the pathophysiologic mechanisms that underlie product-specific safety signals that might arise during clinical development, what's really left in are the scientific gaps in establishing evidence of effectiveness. And here's where we see the largest scientific gap, particularly for new vaccines and particularly with those based on emerging manufacturing technologies. Because of the challenges in conducting an adequate and well-controlled pivotal clinical efficacy endpoint study for H5 vaccines during the interpandemic period, we are largely left with having to use surrogate endpoints. And the most obvious surrogate endpoints to establish inferred vaccine effectiveness are immune response biomarkers. Herein is the issue: how to link that immune response biomarker to clinical efficacy. The most straightforward way, as depicted on the leftmost part of this slide, is through inference from seasonal influenza vaccine efficacy. This then leads to the largest scientific gap from a regulatory perspective, and that is for those new vaccines for which efficacy has not been demonstrated for seasonal influenza with the same manufacturing technology using the same process and the same formulation. This immune response biomarker gap is generally the one that has engendered the most discussion with vaccine manufacturers about producing or even updating vaccines against influenza, H5 influenza, the details of which I'm not at liberty to discuss in this setting. So let me briefly mention just one other gap, which is the programmatic one. It's not really a scientific one in H5 vaccine preparedness, and that is evidence in special populations. And the ones most likely to carry the heaviest disease burden during a pandemic. These are persons with immunocompromising conditions, pregnant and lactating individuals, and the youngest and oldest populations, to name a few. Here again, fit-for-purpose immune response biomarkers are often the scientific gap. So maybe to sum up, a couple of critical things that should be done now to improve our readiness. First, highly efficacious seasonal influenza vaccines with demonstrated effectiveness across all high-burden subpopulations, and based on manufacturing technologies that can actually match the response speed needed for a pandemic. And not just the manufacturing time, but the time from the factory floor to functional population-based protective immunity. Second, fit-for-purpose, analytically and clinically validated surrogate endpoints, likely based on immune response biomarkers. And third, line of sight to a letter of regulatory approval, rather than just to a letter of authorization for new or updated vaccines. That is, line of sight to approval rather than line of sight just to emergency use authorization. And so with that, I think I'll turn it back to you, Norman. Thank you.
Thank you, David. Thanks for those comments. And now we'll get a perspective from, and I'm sure this is generating a lot of questions in which I think we'll have time to get into, but we want to get a perspective from Europe. And our next speaker is Marco Cavaleri from the European Medicines Agency. Marco, I'll turn it over to you.
Thank you very much, Norman. Find this slide. So yeah, indeed, I will give you a perspective from the EMA, and you already heard a little bit from Professor Monto what we have been doing in Europe in 2009 when we had the mock-up concept, how we called at that time, in place. So, a pre-approved vaccine built with an extreme case in terms of a very distant influenza A strain, H5N1 was selected at that time. And then we used that as the basis for a rapid approval of a pandemic vaccine, simply changing the antigen and without the need of waiting for any clinical data, either immunogenicity or safety, before this change could be introduced. Now, in the aftermath of the pandemic, we adjusted that plan because, indeed, as said, we need to have some flexibility. Not all pandemics will be the same. You will not always be in the same position, and indeed, your posology for the vaccine might change, and therefore you need to have at least some data that could guide you in deciding which route to take in that type of scenario. But also, let me say that the adjuvanted vaccine that we approve in Europe turned out to be more effective than the non-adjuvanted one, particularly in young age group and in children, where indeed the human population tended to be more naive to influenza in general. But the first point that I want to make here on this slide is that, beside this approach of approving what we call now a pre-pandemic influenza vaccine that is just put on the shelf, is not marketed, it's just for the sake of having a backbone that we could use and, really change through the antigen and the approval when a pandemic strikes. We also have another regulatory pathway, which is the one that is for zoonotic influenza. So this pathway allows us to formally approve a vaccine that could be marketed differently from a pandemic preparedness vaccine, and is really aimed at protecting frontline workers, veterinarians, or whoever might be exposed to the risk of being infected with an avian strain of influenza. And what I'm showing here is the most recent approval that we gave this year for a vaccine from Seqirus, which is based on the H5N1 strain that is causing this animal pandemic, as previous speakers mentioned, and is based on this new clade of H5N1. Because even if we approved already vaccine based on the same construct with the previous version of H5N1, we know that cross immunity is not great. And therefore, it's important that we are matching what is circulation in order to be sure that we have an immunogenicity profile that will make us more confident that could be an adequate level of protection in case people will get infected with this strain. And you see here on this slide the type of indication that we have been giving for this vaccine, which indeed was built with the H5N8 Astrakhan. And indeed, the scope is for active immunization against H5 subtype influenza A viruses in adults above 18 years of age, which is another major difference with the pandemic preparedness vaccine, which they are approved straight away for the entirety of the population from birth, while here is more focused. And also there is a need of bringing a quite adequate package of safety data when it comes the time to approving this vaccine. But of course, also here, we took advantage of what we already knew about this platform and the previous version of H5N1 that illustrated the ability of the vaccine to be adequately immunogenic and safe. And therefore, for this approval, we do not require any safety of immunogenicity data because we felt that the antigenic difference was minimal and did not require, to have a proof pre-approval that the immunogenicity was as expected. And then moving to some more general consideration from the European perspective, as said, we got pathways for regulatory approval both for zoonotic influenza and for pandemic preparedness vaccine. And so far we approved, or at least we have still approved in Europe, 3 zoonotic influenza vaccines and 4 pandemic preparedness vaccine for the time being, based on a different type of construct, including a live attenuated influenza virus that is pre-positioned as a pandemic preparedness vaccine. And we're looking forward to looking to expanding our armamentarium and making sure that maybe in the future we will have even more type of vaccines that could be included here for the sake of preparedness, or to have options to tackle zoonotic influenza. Some of the consideration that I have to make in addition to this, general presentation is that what we know already now, and we had some discussion with manufacturers right this year, is that even, many years down the line from influenza 2009, H1N1, still the timeline for updating the manufacturing and having batches ready to go for vaccination campaigns with this more traditional protein-based vaccine is up to 6 months or even more. So this is quite a long time frame, and is a bit frustrating, I have to admit, that we have not been able to come up with ways of shortening these time frames and making sure that we could have these vaccines available much earlier than that. And one of the pitfalls, resides, as David also mentions, in the potency assay, in the availability of reagents for, testing the potency of the batches, and then coming up with a rapid decision then on what would be the right way for manufacturing and approving this vaccine from a CMC perspective. So this is an area that is still a gap, and we need to work on that. And, the fact that it will take at least 6 months to have this vaccine ready to go raises the question, can we use platform technology that are much faster than that? And of course, we went through the COVID-19 pandemic, and with new technologies such as messenger RNA vaccines, we know that from a manufacturing perspective, they can be much faster in coming up with batches that can be released. Of course, I take knowledge of what also was said here in the chat about the equity, the possibility of having large amount of messenger RNA available for very large vaccination campaigns, but is without doubts that messenger RNA vaccine can be much faster in delivering at least first batches of vaccines that can be approved and then used for vaccination campaigns. The problem, as you know, is that we don't have yet any messenger RNA vaccine approved for use for seasonal influenza, and we are still navigating into how to come as early as possible with any approval in this direction, and how to build then an approach that will look into immunogenicity and course of protection in order to make sure that then we can come out with a rapid decision if a pandemic strikes in authorizing these vaccines without waiting for clinical efficacy data. So this is extremely important, and we are working on that. We are working with developers and the international regulatory community to make sure that we are doing our best to be as prepared as possible in this direction. And of course, safety is equally important. Vaccines have to be accepted by the population. At the moment, there is a big wave also of miscommunication and anti-science movement that are going against messenger RNA vaccines and their safety. And that also is an important area that we need to tackle for the sake of preparedness. And also we are dedicating time and effort in this area because we do believe is equally important. And the last bullet here is what I already started to mention, that we need to have better assays for understanding the immunogenicity of any new vaccine that we may think about approving and using in the context of a pandemic caused by influenza. Probably it's time to think about other assays that the hemagglutinin inhibition assay, and to move into microneutralization or neutralization assays or any other type of assays that could be reliable and could be used across the board, also with different platform technology, to be used as a course of protection in deciphering and understanding to what extent this vaccine could be protective. So that also is an important cross-cutting area that we would like to see more work to be done, and we are ready to engage with the scientific community in this direction in order to be a very strong position about any possible approval of a pandemic vaccine based on immunogenicity only. So I will stop here. Thank you very much.
Thank you, Marco. And the last speaker in this session, Allesandro from CEPI, and I'll turn it over to you.
Can you, thanks, Norman. Can you see my slides? Can you see my screen?
Yes, great, thanks.
All right, yeah, thank you all for joining and the organizers for convening a meeting on such an important topic. So today I'll be speaking on behalf of CEPI. So CEPI is the Coalition for Epidemic Preparedness Innovations. And while influenza isn't typically part of our portfolio of priority pathogens, our work in vaccine development more generally has given us a unique insight into some of the global regulatory challenges that we face in preparedness. And what we're seeing in general for the influenza space really mirrors many of the challenges we face in our other programs. So I'll be addressing three key areas today, examining both the challenges and potential solutions for each. I'll start off with some of the regulatory pathways. I'll talk about evidence generation, and then finish off with a little bit on equitable access. So let me start with the regulatory landscape. We're facing three significant challenges here. First of all, while David and Marco highlighted how the US and EU have developed robust mechanisms for pandemic influenza response, including specific pathways and really working with developers to address these, many national regulatory authorities in lower and middle income countries really face resource constraints more across the board. And this really limits their ability to prioritize preparedness activities, especially for diseases like influenza, where maybe are at the bottom of their list of priorities. Second, as we saw during COVID-19, and this isn't yet fully resolved, many countries lack legislative frameworks for emergency pathways. And third, while many low middle income country regulators rely a lot on WHO guidance, the current guidelines for pandemic influenza vaccines, including the TPPs that are out there, don't fully address novel technologies that have been made available, for example, during COVID-19, like mRNA, but others too. And this creates particularly challenges for countries depending on these guidelines to roll out novel technologies or potentially apply them. So how do we address these challenges? Well, first of all, we really need to kind of work on enhancing regulatory capacity buildings across the board, particularly focusing on applying reliance mechanisms, especially in pandemic settings. We must work directly with countries to incorporate emergency use pathways into their legislation. And finally, we should advocate for updates to all relevant guidance to include considerations for novel technologies, particularly their use in LMICs. And I know agencies like FDA and EMA are updating their influenza guidance to include some of these novel technologies. So it will be important to roll that into broader discussions. Then let's move to the second area, which is around efficacy and evidence generations. Here are the key challenges we're really facing. The first is the limited use of seasonal influenza vaccines in low and middle income countries. And this has really created two significant gaps. As both David and Marco said, a lot of the pandemic preparedness elements of influenza really are focused on leveraging data generated through seasonal influenza, both in terms of strain change, but also in terms of clinical data. So we have limited, in a lot of low and middle income countries, we have limited data on vaccines efficacy and safety, especially in the special populations. The combination of limited usage and lack of influenza-specific pathways creates general uncertainty about what a regulatory requirement would look like. And this is emphasized even more when we're talking about novel technologies that are not even out there yet, such as the mRNA ones. And this might be that might be deployed in an emergency. I mean, WHO has the pre-qualification procedure. If we take that as an example, there's only currently one H5 vaccine that is PQ'd, and that's only indicated for a pediatric population. So again, how would things look like if we needed to quickly move in terms of approving? It remains a bit unclear. Now, to illustrate this challenge, during COVID-19, we saw how critical local efficacy data was for vaccine acceptance and uptake. And in several African countries, the lack of locally generated evidence created barriers to both the regulatory approval and more broad public confidence. We can't afford to face these same challenges with a potential pandemic influenza vaccine. Therefore, to address these evidence gaps, we would recommend continued investment and support in existing surveillance systems, broader evidence generation focusing on novel correlates of protection, but also on novel technologies, especially applied to the specific populations, and then research initiatives on these technologies, and again, focused in LMIC settings and on the populations that would be impacted in a pandemic. Finally, if we talk about the technological aspect, and both our previous speakers, but also from other sessions have talked about the technological innovations that are ongoing. There are two real issues if we take the perspective of global health and low and middle income country settings. First, many of the technological innovations that are being tested in high income countries are really adapted for high income country health systems. Making this less suitable for rapid response in low middle income countries. This is reflected in the small proportion of clinical trials occurring in low middle income countries, which, as mentioned earlier, limits evidence generations, limits potential trust of using these vaccines in case of an emergency, and more generally limits the vaccine availability. Second, as mentioned by both Marco and David, again, the requirements for potency assays and the speed at which these could be developed and available really limits the speed at which a vaccine can be rolled out, especially in countries which at the start of anything have limited or no manufacturing capacity or no kind of broader importing or exporting capacity. Now, so to address some of these challenges, what can we do? So first of all, really ensuring that ongoing developments includes specification relevant to low middle income countries, such as thermostability or needle-less delivery. Now, CEPI, just to give you examples, it has multiple projects working on achieving these two outcomes. Then we must really support the implementation of the World Health Assembly Resolution 75.8, which was to enhance kind of clinical research capacity around the world, to build these clinical research capacity sites. And this is a very general topic, but actually we see that building this clinical research capacity is critical to support broader pandemic response efforts. And this includes regulatory systems around the clinical research. And then finally, developing and adapting alternative potency assays to accelerate the vaccine availability, particularly in emergencies. So yeah, looking ahead, we see real opportunities to transform how we approach these challenges. The innovations we're seeing in vaccine platforms, especially mRNA, but other technologies as well, could really drastically accelerate how we respond to pandemics, but only if we also address the regulatory issues that are ongoing. And even though high-income countries like the US or EU are really thinking through these regulatory challenges, in other countries where there are other health priorities, these sometimes cause a real problem in terms of rolling out any innovations, especially in terms of a public health, in the case of a public health emergency. And so in CEPI, we really advocate and work with our partners to advocate for a systemic global approach to vaccine development and deployment. And I think I'll just stop there. Thanks.
Thank you, Alessandro. And thank all the speakers in this session. We can move into some questions. There are a number of questions that have come in, and so we can move into that. We have time. I'd like to start with a question, and I'll present it to the entire panel, but David, you may want to start, or we'll see. But if the recommending bodies such as the ACIP wanted to make a recommendation that high-risk populations be use one of the currently licensed vaccines for H5, what are there regulatory and scientific challenges or gaps that would preclude the use of the currently licensed vaccines?
Yeah, so Norman, thank you for the question. I don't know if there are any regulatory gaps. As you know, part of what ACIP does is to look at the approved vaccines and make recommendations, sometimes off-label in terms of how they're used. But I would make a call out that even for routine vaccines, we have lots of evidence gaps in the special populations that I called out, be it pregnant and lactating individuals, immunocompromised individuals. And really, I think part of the goal should be to generate evidence that can be put in the USPI. So healthcare providers and the consumer have access to that evidence in addition to ACIP. So what I would say is it's a general evidence gap for vaccines that I think more investment is needed to fill over.
Thank you. Any other panelists want to address that? I mean, Marco, but it's somewhat different in Europe, especially given some of the things that you just presented. So you want to comment on that? Whether the recommending bodies, I mean, for instance, your country, you go ahead. Go ahead.
Yeah, no, indeed, because we have these options of approving a vaccine for, zoonotic influenza, that is a vaccine that can be commercialized straight away. So we approve it, and then it's up to the member states, NITACs, to decide, whether they want to use it or not. And in Europe, for example, Finland has decided not only to purchase this vaccine, but also to administer it to frontline workers, like, in the mink farms or in other farms in Finland, while other member states, opted for just stockpiling this vaccine and to keep it on the shelf and to decide later on if they want to use it or not. So this is a pathway that would allow flexibilities in the member states on how they want to use it and what kind of use they want to make it of this vaccine in a pre-pandemic setting. Completely different is the situation for the pandemic preparedness vaccine, which are vaccines that are not commercialized at all. They're not aimed for really being marketed or stockpiles, but really just a concept to allow rapid regulatory decision in case a pandemic strikes.
Thank you, Marco.
Can I just
Uh,
ask, I'm just
John?
curious, Marco, will Finland be doing any vaccine efficacy or effectiveness studies in those farm workers who are being vaccinated?
Yeah, I think there will be a session. Anna, okay, I see you there. Anna, over
Yeah.
to you.
Hi, Mark, and hi, everyone. Well, we'll discuss that in next session. So let's save the questions there.
Great. And another question I would ask is, there seems to be a lot of activity for the mRNA vaccines moving forward, but what's being done, I mean, for vaccines that, especially thinking about the developing world where the cost of these vaccines may be unaffordable. And so what's being done for other maybe somewhat slower, but more cost-effective platforms that can be used throughout the world? And maybe John or Arnold or any other panelists can address that, as well as Allesandro.
I mean, CEPI plays a small part in addressing a lot of these challenges. We're a funder, an R&D research funder, and a lot of our call for proposals have focused on how to develop these technologies in such a way to address some of the needs of low and middle income countries. And so it's ongoing. There's a lot of activity in research at very early non-clinical stages even, but we're not there yet. So if tomorrow there were to be a pandemic, I think we would be relying on the things that are out there already. And so it's really trying to question of how do we ensure that whatever is available now, can we get that to everybody that needs it? I think that is still a problem and still an issue we need to resolve.
Thank you. Other panelists want to comment on
Well,
that?
I think there has been an effort to try and establish vaccine manufacturing in countries that didn't have that before. This wouldn't be a new technology, but just to expand the sort of base of traditional flu vaccine manufacturing in other countries to try and have more availability of those kind of options.
In the past, there's been an effort to look at live attenuated vaccines because they can be produced quickly, and the technology could be made available in developing countries. It's not nearly as complicated. The problem there is the evidence basis for live attenuated vaccines being useful in this situation. Over.
Thanks, Arnold. Any other comments on that? If not, I'll move to another question. Since HAI is sort of the biomarker for our current influenza vaccines, how can this be used as a surrogate for RNA vaccines? And you mentioned, David, the challenges of this, and my assumption is that those vaccines will currently be required to do efficacy studies. But how are you guys considering, or are you considering the HAI for a surrogate for those vaccines, especially since that platform has been at least demonstrated in other vaccines, COVID, as far as effectiveness?
Norman, maybe I'll take the first go of that. And maybe to answer that, point to two current guidance documents. So the first one is expedited programs, where there's a lot of discussion about surrogate endpoints reasonably likely to predict clinical benefit. And I think what you'll see in that guidance document is the criticality of those endpoints being on the causal pathway, or at least some correlates with clinical outcomes. And so I think the other one that I would point to, I know we're talking about H5, but the current guidance document on development and licensure of vaccines to prevent COVID-19. And there, I think we point out that it really depends on the vaccine characteristics, and a potential surrogate endpoint would likely depend on the antigenic structure, the mode of delivery, the antigen processing and presentation in that individual vaccinated. And I think we point out in that guidance document that an immune biomarker established for one vaccine type, in this case, we might say inactivated virus vaccines, cannot be presumed to be applicable to, let's say, a nucleic acid-based vaccine, given that the two vaccines present antigens in different ways and they may engender different types of protective immune responses. So what we really want to see is the totality of evidence that suggests that that biomarker, that immune response biomarker, is actually applicable to that other manufacturing technology. Over.
Thank you.
Yeah. If I can add, I think the question is very pertinent because, if you got a messenger RNA vaccine that, is the sequence and the translation is for the full HA, you may wonder, what other type of immune response would it generate? Why would it be so different than more traditional vaccines? And this is something that we are heavily debating within the EMA as we are redrafting our guidance for influenza vaccine. But there is also the other element that if a messenger RNA vaccine company is interested in having an approval for seasonal influenza, we still think that generating efficacy data might be preferable, as a way of providing good evidence that would also support the benefit risk balance and, decision by NITAGs and recommending bodies. So it's under discussion at the moment. I still think it's plausible. But of course, we need to discuss this more and also with the scientific community to see to what extent we can go straight into this type of, antibodies, functional antibodies to the HI component. And as I said before, we also would like to move away from the HAI assay, which, as you know, also the red blood cells that you're using are influencing the outcome. And also during last pandemic, we had a lot of issues, at least with some manufacturers in this sense. So really, having data set also from different type of assays that measure functional antibodies, such as microneutralization with different incubation period to try to capture all kinds of antibodies, might be helpful to corroborate the fact that we have a good understanding of the immune response that could be protective here, and considering using that also in the context of the messenger RNA vaccine. So to summarize, I would say this is work in progress for us, but we are not excluding this option, particularly in a pandemic context.
Just to kind of raise one last point in the context of testing is similar to what I mentioned earlier about the need for capacity building in terms of regulatory capacity. We're talking about analytical capacity in a lot of these countries. There's a big gap there too. So anything that gets developed or anything that is new will need to be accompanied by the appropriate training and availability of analytical testing and expertise in regions or in countries where that goes. So again, if we're talking about the way that we roll out these vaccines, there's so many other issues that need to be addressed rather than just the availability of the product.
Thank you, Alec. Another question that came in, and this is for you, David. The comments about special populations or at-risk populations, and it's aimed at the safety database. With the safety database size of, say, a prototype for each of these groups, say pregnant women, children, immunocompromised, what have you, would that be sufficient for these groups for the same indications for pandemic evolved strains of the same type, or would additional safety data be required for each group if or the pandemic strain?
Yeah, no, that's a really good question, and one that we're actually having a lot of internal discussion on. In part, sometimes it's challenging to generate the data in certain populations with a pandemic influenza vaccine. So what I might have to say, which I know is not a satisfactory answer, is, it's on a case-by-case basis. It really depends on what did we see in the safety database with, let's say, the seasonal influenza, and how much of that do we think would translate into for the pandemic vaccine. What I would say again and repeat again, our safety databases for some of these special populations are pretty small, and I think we should be investing more in studying these vaccines in those special populations in order to build up the safety database for those. And so, again, a call out for generating both the safety and effectiveness data in these special populations.
Thank you, David. So from, I guess, for you, John, how is BARDA preparing for this, and what are those needs from their perspective as far as moving these vaccines forward? John.
BARDA is currently supporting clinical trials to establish the safety and immunogenicity of the vaccines that are currently licensed for H5. So studies looking at the H5 vaccine in eggs, or the cell culture H5N vaccine adjuvanted with AS03 or MF59. It is also doing studies to assemble a database to support the use of the egg Sanofi vaccine as well in terms of the H5. Now, BARDA also has a development project for mRNA vaccines, a number of different mRNA vaccines that are in development. But those would probably be a little bit different because they haven't been shown to be effective for seasonal influenza yet. So the pathway forward there would be a longer, more prolonged one. But there is an interest in having that as a platform that could be used for pandemic response because of the attractive nature of the speed with which such a thing could be made in response to a new variant. So that's where things are right now. All those projects are underway. And there is also a large manufacturing establishment that can make vaccines if needed, depending on the circumstances.
Thank you, John. And maybe, Allesandro, you can comment on, I mean, you mentioned in your slides about surveillance. I mean, what are the, what is CEPI doing, and how are they contributing to looking at surveillance of influenza, and particularly, is anyone looking at surveillance of H5? What do we know what's going on? And I think others can comment on that question as well.
Well, yeah, like I mentioned at the beginning, influenza is not typically one of our priority pathogens, so we don't have active programs in that space. And we also, although we have a lot of our colleagues that are working the epidemiology on a lot of our priority pathogens, in terms of influenza more specifically, we have less activities in that space. I mean, what I would say is again, one of the challenges that is obvious is the lack of use of or limited use of influenza vaccines or seasonal influenza vaccines in a lot of these parts of the world, combined with just general more limited resources in terms of doing your safety evaluations or follow-ups is a challenge, and therefore any kind of trial, any research or clinical research capacity that can be built in these spaces is something that we're looking at, and we are investing in various different projects to build that capacity in some of our priority pathogens, but that can then also supplement a lot of work that might be required for influenza. But yeah, I think it's a challenge, and when these diseases are not necessarily on the priority for a lot of these countries, when influenza is not a priority for a lot of these countries, I think it's yeah, there's limited that we can do with the little bit of resources that the systems have.
Thank you. Others want to comment on that?
Well, if I can say on the surveillance side, because there was also a question in the chat about narcolepsy, for example, is that, and also referring to what Dave was saying about, different vulnerable groups. If you have a vaccine that is exactly the same one used for seasonal influenza, you can collect a lot of data pre-approval. But if the vaccine is different, for example, it has an adjuvant such as a ESO3, well, that is more challenging. You cannot really collect data pre-approval. So, and what you can collect pre-approval is too small to define what is the actual risk for rare adverse events such as narcolepsy or other GBS or others. So in those settings, what is really important is that we got the infrastructure that as soon as a pandemic vaccine is rolled out, we can establish where there is any undue signal of safety coming up and having the infrastructure ready for do that, because I cannot see what else we can do in the pre-approval phase to overcome all these aspects. So really, you need large number. You need to have a system that is able to spot whenever something is emerging and to inform the population as soon as possible about any rare but serious adverse event that may occur. And this is, across different vulnerable groups, including pregnant women, children, young children. So that's something that I fear, cannot be fully addressed pre-approval and requires really infrastructure for post-approval studies in the context of mass vaccination campaigns.
Thank you. So as we try to prepare, I mean, in influenza preparedness, what are we hearing, especially from the regulators, from the vaccine manufacturers? I mean, are they prepared? And I know you can't speak for them, but are they prepared to move forward with developing H5 or other pandemic vaccines? What are the gaps that you see that they have? I mean, we don't have anybody here on our panel from the manufacturing side. Or even Arnold or John. I mean, what are you hearing?
I was going to turn to both of them. I think they probably have information they can share.
Yeah.
Well, I can tell you that BARDA has contracts with all three of the vaccine manufacturers that would allow, if it were necessary, a scale up and production of vaccine in the event that there was a reason why that was needed. So there would be a rapid response, and again, unfortunately, the reality of vaccines are that to actually make enough to vaccinate everybody will take time. But there are mechanisms in place that would allow that to be started rapidly if necessary.
I don't hear a whole lot from the manufacturers except they would like a little more guidance in terms of not simply the regulatory process, but the other aspects of getting something prepared which may or may not be used. I'm basically an outsider on this because in part if I get too involved with the vaccine manufacturers, I'm in conflict for my activities on the VRBPAC for FDA. So it's a bit of a problem, but I think participation and of the manufacturers is vital here, because it's a question of their priorities as well as the priorities of public health. Over.
Thank you. And there have been comments, David, you've made some comments, Marco, you've made some comments about, for the most part, the CMC, other than the assay development, has been rather straightforward. But and we've been struggling with the potency assays for many years now. What are the agencies doing in collaboration with the manufacturers and others to really resolve this issue? I mean, this could be a gap, a bottleneck, And so where are we now and what's being done? Do you foresee us resolving this issue anytime soon?
Well, if I can start, I don't see this as being resolved anytime soon, and I don't think we have done enough, to be honest with you. You know, it was on our list after pandemic 2009, and one of those areas that required attention and to come out with a new strategy to get away from the current potency assay, think about, you know, HPLC and other options. But I don't think this agenda has really progressed. So it's something that we should take the opportunity to go back to that, work internationally, work with the manufacturers, and try to figure out what else we can come out with that could be a suitable alternative that we make regulators comfortable in taking a decision on the potency of the batches, and at the same time streamlining this entire process so that these more traditional vaccines can be ready for deployment much earlier than what is the case right now.
Yeah, I'll just echo that. I mean, we're in the 21st century. We should be applying 21st century technologies to this, but that takes up quite a bit of an investment to get those analytically and validated. So I think there's a need for investment there and a need for bringing to bear newer technologies that are available.
And we got another question about to what extent are there data in support of the 1 to 40 titer or any other threshold for, say, an H5N1 vaccine? So as far as a protective titer, I mean, are there data there that would support that?
I don't think we have data to support that, and that's why we abandoned this across the board. You know, the 1 to 40 titer, you know, as you all know here, comes from, you know, long time gone times when human challenge models were done in the UK. So with 50% protection, moreover. So, you know, I think it's really time that we look at something different. We really need to change the mindset and to really try to figure out which immune marker would be best suited. But it's true that in the pandemic context, it's a challenge because, you know, we may think about having microneutralization, let's say, as the preferred option with different formats on top of HAI. But then, you know, to think about a threshold is not possible. I don't think we have one. And we would really like to work here with the scientific community to advance here our understanding, or at least of what else can be done to come out with some ideas on how to interpret all these different immunogenicity data, because this could be quite a struggle with traditional vaccine. And of course, it will be even more difficult for non-traditional new antigens, which is another story. But there were some questions on the chat on that as well.
Okay.
Well,
I think this is a major issue, that try and come up with a better correlative protection, especially when you realize how the HAI assay, you could do that test in 10 different labs and they're going to give you 10 different answers. So it's good because having that antibody does seem to be associated with protection, but trying to quantify it, it's just,
We've actually published papers on how the 1 to 40 titer works when it works and doesn't work when it doesn't work, and how because of prior immunization and things like that, it doesn't really work very well in predicting efficacy. I invite people to look at the Hobson paper to see how weak the data are in that paper itself, on which we are relying. Over.
Thank you. And I want to, we've run out of time. I want to thank the panelists. This has been a great discussion. I think we have a better idea of where things are, and it's from a regulatory research and development process, perspective, as well as from global. So again, thank you, everyone. And I will turn it over to the organizers.